This is episode 85 of the Immunology Podcast, Reproductive Immunology with Dr. Adrian Erlebacher. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Adrian Erlebacher from the University of California, San Francisco on the podcast to talk about his research on the immunology of the maternal-fetal interface. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
Are you working with mouse immune cells? Why don't you get a free wall chart of the estimated frequencies and percentages of more than 25 immune cell types in mice? To learn more and get your wall chart, visit stemcell.com/mouse immune cells. I have the one for the human cells. It's quite pretty.
is it next to your lab bench?
Well, it's next to our desk area. The lab has not a lot of empty wall space.
Ah,
Not even for this beautiful chart.
but do you guys have desks in your lab or in a separate area? Because our desks are always next to the lab bench.
I know that's a very American thing, but here I think in Europe you're not because you're not allowed to do anything in the lab and you cannot have a cup of coffee. So they tend to separate them.
You're not allowed to
So
have that either. You're just not supposed to have coffee at your desk, and everyone ignores that because it's ridiculous.
exactly, that's the thing. I would also want to have a coffee at my desk, so that's not compatible with having my desk in the lab.
No,
Um,
difficult.
Never have I done that, nor will I do it again. But no, here we have separate.
So, spending time in the lab, you've gotten out at all? Any movies or anything else in life in the summer? It's blockbuster season.
It's blockbuster season. Well, we went to the movies a few months ago, and we were like, starting was this indie movie, it's just not like a small a Chilean production, and then like 3 minutes into the movie, there was a citywide power shortage, which never happens in the city, and so we had to leave and we got refunded. So yeah, that's so much for my movie.
You said in the city, do you have power shortages in the country ever?
Nah, I've never. I don't think this happens very often. Was
Oh, so
very.
something must have blown.
Yeah. I think there was a reason. There's some transformers blew up. I don't remember exactly what happened, but it was so weird because that never happens. The first time I heard that, you had I don't know, like a whole area of the city was without electricity, and that's where and we decided on that very day to go and watch a movie. How unfortunate.
I have seen Deadpool and Wolverine and highly recommend it for the stupid fun that it is.
I can
It is not
imagine.
for deep thoughts. It is for
Oh no.
low class entertainment of the best variety, and it's amazing. Especially if you're a fan of Deadpool or Wolverine or Marvel in general. As Deadpool says, he is Marvel Jesus.
I see. I see. I like Ryan Reynolds. He's funny.
The movie is absolutely ridiculous, and Chris Evans has the best cameo ever.
All right. All right. Listeners, if you want to go watch a movie, go watch Deadpool. I will consider it. I think, is it like, can you watch it like in 3D IMAX
You can do it in
experience?
3D, IMAX, upside down, backwards, whatever. But like, it's worth seeing in a theater because it's just hysterical and funny on a big screen, and you know,
Okay.
it's a good time. It's a good 2 hours. You'll laugh a lot. It'll put you in a good mood. So when your experiments don't work all day long, and you go home, go see Deadpool.
I could use some Deadpool some of these days. But here in Amsterdam, summers are for taking your boat to the canals and enjoying, bringing some beers and drinks, some non-alcoholic beer. We have excellent non-alcoholic beers, by the way. It's amazing the variety nowadays. And you just go to the little 600-year, 600, 500-year-old canals in Amsterdam.
You have a boat?
We do. A lot of people here. Actually, I have to say, I admire that of Amsterdam. It's such you can have those little luxuries are fairly accessible. And it's very nice. A lot of people have. A few of our friends also have
Do you guys
boats.
water ski at all or anything, or tow people
No, no,
behind?
here is like max speed is like 6 kilometers per hour. So like walking. Imagine if you're going
I mean,
water skiing in a canal. you would just crash against the next, tourist boat, a touristic boat in the canal. It would be complete madness.
Sounds like a challenge.
Sure, sure. Don't. That's so American. Do you water ski? Do you bring? How is it called? Those
Jet skis?
jet ski. Do you jet ski in
I
Amsterdam? Of
mean,
course
we
not.
we
Of
have a
course
boat
not.
and we jet ski. Well, not a jet ski. We inner tube and water ski.
Yeah. Now here's different. Here we bring snacks, drinks, friends, conversation, and we just aimlessly wander across the canals.
Yeah, you do that at the lake, and then when you're done and you want to do more, then you just water ski, and then you go back to doing that.
Yeah, no, by the time it's like we don't that part we skip. We just do the slow life part. But I don't know if people do it that a lot. I think all the waterways here is a little bit tight for that. Yeah, I think people only do those kind of things in the like by the sea. People do a lot of kite surfing. Kite surfing is very popular.
I've done that once.
Mm. I haven't done it. Sounds looks very fun.
The wind pulls you on your face the first few times.
I can't imagine. That's why I haven't tried. Oh well. Anyway,
All right.
enough of this. We should get down to the business. What are you going to talk about today?
Well, I'm going to talk about your brain, but not your
My
actual
brain?
brain.
I
I'm
know
going to
my
talk about
brain.
your skull, and not your
Ooh.
skull, but the immune cells in your bone marrow in your skull.
Wow. Okay. You have my attention now.
All
Tell
right.
me more.
Title is Cranioencephalic Functional Lymphoid Units in Glioblastoma in Nature Medicine, published the 31st of July. First author is Celia Dobersalki, and last author is Bjorn Scheffler. All right, so first up, guess what, folks? You have bone marrow in the skull brain. Right, so bone marrow in your skull. And the question was, well, the brain is an immune privileged organ is the classical view. Glioblastoma has a real problem because as we see when we plop out immune cells, the immune cells for glioblastomas, immune cells seem pretty senescent and exhausted and immunosuppressed, right? So that that not activated phenotype, otherwise you wouldn't have glioblastoma, right? Because your immune cells would kill it, and they don't. And that is true in this paper and elsewhere of what's going on in the tumor. But where do these immune cells come from? What's going on? What about this compartment, which has never really been looked at? So they look at treatment naive glioblastoma tumors and at the cranial bone marrow of people. And they also take as a control group from patients. This is all patient samples. Non-malignant intracranial disease as a control. And they do clinical and anatomical imaging, single cell molecular studies, and immune cell profiling, quantification of tumor reactivity by CD8 T cell clonotyping. So what they find, and this is really just the kicker, is that the marrow in the areas around your marrow in your skull have tumor reactive CD8 T cells. Way more of them than in the tumor itself. And in the part of the skull near the tumor, as opposed to a more distal part, a part further away from it, they have increased reactor of these effector types and more expression of the lymphoid egress marker S1PR1. What's weird, though, is that, and these cells do leave and go to the tumor where some of them get suppressed. But they then also see that the more of these you have, which can serve as a marker of focal association. So the more immune cells you have near the tumor that are active in your skull, you have increased risk or increased likelihood of progression-free survival. So one of the suggestions they're looking at is, could you use a CXCR4 radio label, right? So just like you do for regular PET scan, but could you use that as a way to predict outcomes of glioblastoma? Because it seems to be the more activated immune cells you have in the skull marrow, the more likely you are going to do well. But really, they see that these T cells come from the brain. They can go to the tumor, but then in the tumor they do get shut off. So the tumor microenvironment's shutting it off. They don't come there exhausted though. They don't come there suppressed. They go there and then downregulate. And they don't explore that. They explore just what's happening in the bone marrow of our skull, and how this is actually apparently super important for cancer outcomes, is how much activation you have in your skull marrow. And there is quite the effector population there, which was kind of interesting. Like, okay, you have regional effects of bone marrow.
Okay. That's, I mean, because it is just, it is not uncommon that T cells will reside in the bone marrow. Memory T cells or activated T cells
Yeah.
will be found in the bone marrow. And I guess the skull is no different.
Yeah. They're just hanging out there, which is kind of unique.
Mm-hmm.
But they seem to really matter for disease outcomes. So they're probably, hanging out and killing stuff that's getting away from the from the tumor, right? Like, you may not be able to, if they go to the tumor, they may get a little too quiet, but they can serve as sentinels to prevent escape of, cancer cells in the rest of the body.
Although it's nice because we haven't really known that long about this bone marrow in the skull, right? We covered some paper some time ago in which they were also looking into hematopoiesis in the bone marrow and maturation of certain immune cells in this bone marrow that was quite also unique. So I think it's very nice to see different implications for the function of this particular bone marrow in the skull to different diseases.
Yeah.
All right. Okay, why don't we then, given that you already started in the head, let's move down to your nose. I think you're gonna like this paper because in some way it's about, mucosa, and it has it all: vaccination, nasal immunization, new cells. Uh, I think it's a very nice story, but it has kind of a big caveat that I want to kind of discuss at the beginning. But I think it's still very interesting. The paper is called Turbinate Homing IgA Secreting Cells Originate in the Nasal Lymphoid Tissues. Published in Nature, 31st of July. First author Jingjing Liu from the lab of Ziv Schulman at the Weizmann Institute of Science. And this paper basically tries to understand, one is the origin of IgA secreting cells in the nose, especially now that there's so much hype or so much interest around nasal vaccination and protection against disease, in the nose and respiratory viruses and things like that, COVID and influenza. We've talked about this a few times already. So they're looking, they're doing some very fancy microscopy in which they look at kind of the whole structure of the nose, trying to understand how it is it that the principally the IgA producing B cells develop, where they come from, how do they find their place in the nose, and how to sustain this production of IgA in the nose. And so I think the big caveat of this paper, if I may start with it, is that something that I learned, when I was reading this paper, is that there's quite some differences between the mouse and the human nasal cavities. Also, in terms of the immune cells that you find and the structures that they generate. So, all of this work is done on murine noses, so on mouse noses. And they're studying this kind of nose associated lymphoid tissue. And there's a lot of debate about, what exactly are the this term means also in humans. In the case of mice, you have the structure that is, this nasal cavity has different areas. But I think the more kind of the simpler way of describing it is that you have a three areas, three bones are called this turbinate, the nasal turbinates. They just these bones that allow for a lot of like surface, a lot of crevices and a lot of surface, and therefore you have all these olfactory epithelium where when you pick up all the scents. And you have, direct connection neurons to the brain for the olfactory signal. So it's also a very quick access to the brain through the nose. So it's a very important area to protect. And so there's one structure that you find in mice, which is this what is properly the nose associated lymphoid tissue, which is something that looks like a non-draining lymphoid structure similar to a Peyer's patch in the intestine that holds a bunch of immune cells with a certain structure. And this is known to be very important for to play a role in in nose immune responses in mice. However, humans don't have don't have a structure like mice have. Inside the nasal cavity, there is no kind of aggregate of immune cells that you can identify that has been identified really in human nasal cavities. We do have our ganglia, so we have the tonsils, so we do have those structure, but those are proper kind of lymphatic organs. They're draining, so they're not necessarily the same as this null structure that you see in mice. So that being said, let's see how mice respond to nasal vaccination in this model. I think it's nice because they have some really nice, very carefully done microscopy. So that's really pretty figures and quite convincing. They show that in mice you have this knot structure that looks a lot like a Peyer's patch, and they have a system in which they inject the mice with B cells that respond to this NP antigen that it's only gonna kind of B cell only. It's not a protein antigen, so it does not recognize the T cells. But then they do an NP plus OVA stimulation, so they have kind of a full stimulation. So if they treat the mice, if they inject these cells, and then they do an NP OVA intranasal immunization together with an adjuvant, they show very nicely that they can follow the development of the immune response, particularly of the B cells, and they show that in mice, this immune response starts kind of around 3 days after the inoculation or the intranasal inoculation, and there's this nult is where the B cells and the T cells start kind of clustering in the structure in the nose. Then they are kind of generate bigger. The B cells generates large clusters in proximity to a structure called the subepithelial dome, which is an area also found in Peyer's patches that has kind of a high amount of CD11C positive cells, so potentially antigen presenting cells. So the B cells, these transgenic B cells recognizing this epitope, they are going to this area, and this is where they are proliferating. And eventually they move to generate germinal centers away from this subepithelial dome. And this is where they kind of generate proper affinity modulated responses. Eventually, these, and I think this is probably the most interesting part, you end up having the plasma cells that make the IgA antibodies end up colonizing. They don't stay in this null structure. They end up colonizing the surface of the turbinates, so of these cavities in the nose. And that's where you find the production of the IgA. And I think kind of this is, in this work, they go through a lot of lengths showing this how the timeline evolves, trying to show that there's this kind of orderly sequence of events in which the B cells get activated, then they get activated by antigen presenting cells in the SCD, then they get generate clusters and germinal centers, and then eventually they leave the node and they move towards this under the epithelium of the turbinates in the cavities. And I think what's most interesting is that in order to move, they don't just like, in order to move from the SCD, from the NALT itself, they actually seem to go back to the circulation and then find their ways back to the turbinates through, and this is mediated by CCL28, that promotes this B cell homing into the nasal turbinate. So basically, what you have, kind of in a nutshell, you have B cells that are fine, that are already have to be pre-existing in the node, because if you have like if you have a completely naive situation, it doesn't really work. You need to have some population of B cells that are already reactive, and T cells as well that are reactive to this antigen. And this is very interesting because this kind of supports the idea that having a priming first with a, you know, regular subcutaneous immunization and then having an intranasal is probably better than having the intranasal vaccination to start, because you need this pre-existing population of B cells. It's not clear to me whether they're already IgA when they start, but this is kind of what they suggest in this paper. And then these B cells, they activate in the node, they generate germinal centers, and then they migrate away and back into the nasal turbinates where they reside and produce IgA. I think it's very interesting. However, again, it's difficult to say how this translates to humans, given that we don't have an alt, at least hasn't really been described with this same characteristics, this non-draining lymphoid structure in the nose. We do have draining like lymph nodes in the area. So for us, what we call NALT, or like nasopharyngeal lymphoid tissues, is different. But it has been described that humans also have, you know, production of IgA in diffuse structures in our noses. So maybe it's kind of similar. So it was very nice to see, like, kind of understand how is the order of events, and understand what is behind nasal vaccination. And I think particularly the need of a pre-existing primed population makes a huge difference when it comes to establishing the IgA producing cells that you want.
Do they at least look at the turbinates in humans at all and see the B cells there? Because humans have plenty of turbinates.
So not in this paper. There's actually not a lot done. But I found a paper. One of the references is this paper from in which they did a post-mortem analysis of turbinates in infants, because also apparently infants have like bigger structures than adults. So, and they see some kind of populations of cells living in the human turbinates as well, but definitely not the kind of nulled structure that you see in mice. But we do seem to also have IgA producing cells living in our turbinates, but it's not clear where they're coming from because we don't have the NALT like mice have. So maybe from our lymph nodes that are in our kind of our throat, or that area close um, to the nasal cavity.
All right. Well, I'm going to continue on our body journey.
All right.
I'm going to the nose area and the lungs.
All right. Moving inwards.
From the
Perfect.
mouth, really, not the lungs so much. We're looking
Okay.
at host and environmental factors shape upper airway microbiota and respiratory health across the human lifespan. So the first authors are Marie Lee Odendahl and Wouter A.A. de Steenhuisingen Pitters. And the last author is Thijs Bosch and Debbie Bogert. And I probably butchered all of those names.
Oh, Marile Odendaal and Wouter de Steinhausen Peters. Clearly Dutch people here. And Thijs Bosch would be the. There you go.
JS is not a sound I know how to make.
Well, that's the very Dutch thing. The I sound is horrible when you learn it. It takes a while until you get used to it. But there you go. You're welcome.
All right. Well, after that tour of pronunciation fail, thank you everyone for watching me struggle live. We'll talk about some microbiota that's interesting. I think this has this does tie to immunology. It's not just a story of bugs, because if remember from the humanized mouse story, if you give a mouse a human immune system, you give them a human microbiota, or a largely human microbiota. So that's a callback to another paper. But this paper, for the first time, we've done a lot of studies on human poop at all ages for microbiota, but less so on other parts. And guess what? Your nose and your mouth have a lot of bacteria in them all the time. Not as many as
Shocker.
intestine, but a lot. And so what they did is they look at nasopharyngeal swabs from people of all ages, plus either saliva for under 10 years or oropharynx for over 10 years old specimens. And they look at just what's happening in all the regions over time. So interestingly, they see that babies have an increase in diversity in the nose as time goes up and as you become an adult. And then after you hit puberty, you're kind of at max diversity. And then, but your biomass goes down. So the amount of bacteria goes down as you get older in your nose. But in your mouth, you start out with a small increase in diversity when you're really young, just a little bit, not nearly this bump. And then it just kind of plateaus and slowly comes down over time in your mouth. And the biomass starts high and then comes down a little bit. There's obviously some confounding here in this group because you have saliva versus oropharynx. So there's a little bit difference. But, you know, first off, this is suggesting that babies are more dominated by less bacteria and they are in high amounts, and that you develop a kind of commensal bacteria over time, which when you see this shift and you think about immune system development, people's immune systems don't mature until post-puberty, really. We have kind of a kids are not adults thing going on with our immune systems, as we've seen from COVID and every other vaccine we talk about, anything else. Like, you're seeing this here with the microbiota as well. Like, you need a mature immune system to reach homeostasis for your bacteria. Then they look at, you know, what's really driving exposure and, you know, what other coordination is going on here, right, besides age. So, you know, obviously they see that you're different niches, right? Your nose is different than your mouth. But the nose was dominated by who you were around. So like you and mommy have some more similar, more so than your mouth. Sex, male versus female, and the season of the year. Okay, allergies. The oral microbiota, on the other hand, was more related to antibiotic use and tobacco and alcohol use. And interestingly, and this is just kind of like a sanity check. If you look at, you know, do you have respiratory systems? They'll see respiratory symptoms. They'll see a clear, distinct shift in the microbiota populations there if you have it. And it's kind of like a sanity check. I don't know why this was such a big deal. I mean, the paper got in Cell, so I think this is part of it, is they were able to do some sanity checks for other things here. But they were able to see that if you had pneumonia, you're much more likely to have strep pneumonia, the bacteria. Right. Like, oh,
Shocker.
Right. But that's why I'm saying it's a sanity check, right? Mm-hmm. If you didn't see that, you would then say, is it just colonization effects that's driving it? But you actually see increases in strep pneumonia with pneumonia, which sounds like the Department of Obviousness being obvious, in particular in the nasopharynx. Mm-hmm. So guess what? Pneumonia, the bacteria, gives you pneumonia, the disease.
All right, but I guess that's not the only conclusion of the paper. That makes it sound like all they did was find pneumonia.
No, I mean, what they did is they did a huge amount of sequencing.
Yeah.
On a whole bunch of people, like thousands. N's are in the thousands, 2,000 a pop. So, yeah. It costs, let's say, $1,000 a sequencing run in a person for microbiome sequencing, at least. This is 16S, right? So it's not whole genome sequencing. So it's cheaper. So maybe 1,000.
So much, really?
Yeah, for 16S for everything. Because now whole genome sequencing is more. Whole genome sequencing is like 4 or 5. for WMS. But even if it's 500, they did 4,800 sample, and they did over 5,000 samples. So even at $500 a pop, this is a $2.5 million study.
Ooh. Well, it does come from the RVIM, I think, is the
Right.
national.
So what it's doing is it's creating the first atlas of this microbiota population as compared to all of the poop that has been studied to date. We've done some work like this. Isn't the first people that sequence something from these categories, but like to have a longitudinal sense of things and what's happening and seeing it track immune system maturation so well is really interesting. Right.
Yeah.
And then the fact that your nose and your mouth, which are not very far apart, have different niches or are significantly different is the other big thing.
All
The
right.
fact that you and your baby have pretty similar microbiome. Everyone knows this. If you've been around a baby, you share a lot.
Fun. Fun. All right.
So I was, when I go on these microbiome papers, I never know what I'm going to get in terms of other cool immunology. I would say this isn't super high on the immunology, but there is some there, especially with this tracking of immune development. And that was really cool.
Yeah.
So, it still gets my vote as interesting science, especially because we've talked so much about respiratory infections and, nasal vaccines and everything else. Well, this all goes together, right?
Yeah. It's all part of the same thing we're studying. All right. So why don't we end up with a bang? Now, as we're moving down, I will go to your favorite organ.
We're going to the colon?
Yeah, actually, yes. Yes, a lot of
Woo-hoo!
what I'm talking about is going to be about colon, or more specifically, colon and Tregs. I mean,
Ooh.
check this out. So the paper I'm going to talk about last is called Chemogenetic Screen Reveals that TRPV1 expressing neurons control regulatory T cells in the gut. Published in Science, August 2nd. First authors Yang Zhu and Kimberly uh, Merskeert, and from the lab of Isaac Chu, which we had in the show at Harvard Medical School. This, as the title suggests, is kind of a neuroimmunology paper, very popular these days, very interesting, and sometimes a little bit out of my comfort zone. There's a lot of kind of neurology things that I don't know a lot, so I have to be googling a lot and like checking out some of the references to understand what's going on. But I think this is really cool because the kind of the what they set up to do is to understand how certain neurons, because there's neurons comes like immune cells, they come in many shapes and forms and functions and markers and different molecules that are receptors that uh, guide their activation so they can respond to different things. And so in this paper, they kind of did this large scale testing of different, trying to understand how different types of neurons can influence the composition or the function or whatever of immune cells in the gut. We know that also the gut has tons of immune cells and they're all very important. And we know to some extent we are starting to understand that the neurons generate, peptides and transmitters and things that can be detected, can be detected by immune cells, and they respond to them, and this affects. So, your brain affects your immune system in ways that are more direct than we ever thought. So, I just want to kind of quickly talk about the model or kind of the mouse model that they use. I think it's really cool. They use this uh, model called DREAD, DREADD, which stands for Designer Receptors Exclusively Activated by Designer Drugs. So these receptors are basically modified G-coupled receptors, G-protein coupled receptors that are designed to respond to designer ligands that are not found naturally in mice, whereas in humans. And so that way, if you introduce the receptors to neurons, they get activated. And they can activate the neuron through can regularly, like a neuron would regularly be activated through a calcium channel and calcium influx, and then this activates a neuron. So if you can find a way to have these receptors activating a specific type of neuron, then you can, see what activating a neuron does to whatever your readout is. That's what they did. So for this, they have a system in which they have a adenoviral, adeno-associated viral a virus, and they use this to infect neurons. This has a neuron tropism in neonatal mice, and then they will only express this receptor if there is a Cree expression. So then they have a bunch of Cree mouse lines for different receptors in or different proteins that are characterized in different types of neurons. And they get in the end they end up having mice, a lot of different mice. So they have 8 different mice with 8 different crees that are kind of responding to the expression of 8 different receptors that are commonly found on neurons, and they're associated that related to their function. And examples, so I think the most interesting example is, which the one I'm going to talk about, is this receptor called TRPV1, which is a nociceptor, is a receptor for pain and for, I guess, inflammation. So they infect the neonatal mice, and then they show that in fact this receptor gets expressed on the cells that are expressing the Cree, that they have this Cree mouse, and then they actually end up activating the receptor and activating the neurons with a synthetic compound called CNO, and therefore they can activate these neurons. So that's kind of the system that they use. And what they show is that they have a bunch of different markers. As I think I'm not going to talk about those because they didn't even give much interesting things, but I think the activation of neurons expressing TRPV1, they do this activation for like 2 weeks in adult mice. And they see that one of the most kind of robust results is that they see a reduction in the percentage of ROR gamma T positive Tregs in the colon of the mice. And ROR gamma T positive Tregs are kind of the most common Tregs in the intestine, and they are usually the ones that are formed upon response to commensal bacteria. So that's usually their main role, and they're usually derived from kind of conventional cells, naive conventional cells that develop into Tregs. And what they show is that this TRVP1 cells, with neurons, can specifically, when activated chronically for 2 weeks, they end up reducing the percentage of Tregs that are expressing ROR gamma T. This is not mediated by differences in microbiome, which could affect the percentage of ROR gamma T positive Tregs, but it's something kind of specific of this neurons in general. They also affect ILC2s in colon, but they're focusing in this, for this paper, for this work, they're focusing on the Tregs. And so how is this happening? They show, and I think this is really cool, they try to see, well, what's happening to the Tregs? Are they dying? Are there different proliferation? So they don't seem to be dying more. They don't seem to be expressing things like RNXin5. They don't seem to be kind of migrating away. And the way they check this is that they have this mouse that has a fluorescent protein called Katie. I didn't even know about this. That is photoconvertible, so it changed from green to from green to red with violet light. So you can kind of like illuminate the colon of the mice, and then you change whatever cells are being exposed, they get changed, and then you can see whether you find these red cells in the circulation later on or somewhere else later on. But well, they use this to show that it's not what's happening. but they see that they have some indication that there's a reduction in the proliferation, and they do this by measuring Ki67. So they think the most likely explanation is that the Tregs are proliferating less. And that's why after 2 weeks, there are less cells than in the control. And this has functional consequences. If they have the mice treated with DSS colitis models, they have more inflammation, the infections like Citrobacter rodentium, which is very commonly used to evaluate Treg function in gut of mice. The mice lose more body weight, suggesting impaired Treg functioning. And they try to understand which exactly are the neuro, and this is where kind of the neurology part comes in. They really care about, there's two different types of neurons that are expressing this receptor that could be responsible for this, and they are they basically show that they have this dorsal root ganglia neurons that are that project to the spinal cord, and they connect to the rest of the nervous system through the spinal cord. And these are the ones that seem to be responsible for this reduction in Tregs, and not neurons that are connecting directly to the brainstem through the vagus nerve, which has also been sometimes associated with connections with and affecting the immune system directly. And this is basically what they show, is that there's a production of a neuropeptide called calcitonin gene-related peptide, CGRP. And this CGRP binds to receptors on the ROR gamma T positive cell, Treg cells. And this kind of generates this negative regulation of the cells. And that's why they proliferate less through this connection. And they use genetic models, and there's a lot of work behind this conclusion. So in general, basically what they show is that you have a direct connection between receptor, like neurons that have receptors for pain and inflammation, and the regulation of Tregs in the colon, in the gut.
Super fascinating. I've seen a talk before this came out here at a conference. Also, those receptors you heard about, that's from a guy, they're called dread receptors, as I'm sure you saw in the paper. And one of my co-mentors, or like on my PhD advisory committee at UNC, Brian Roth, is the one who invented them. And we ended up doing a paper together, not using the dreads, but he also has a whole GPCR screening lab. Pretty cool tools.
Yeah, that was really cool.
They're very neat. They're all over the place now.
Yeah. But I mean, so much work, because I talk about this one, but then there's like literally 7 other combinations that didn't give much results. But so much work to look into this relationships, yep. Right? Very interesting.
They're very cool. All right. Well, we've had some cool science so far, and we're about to have a lot more when we speak with Dr. Adrian Erlebacher at the University of California, San Francisco. But before we get to that, activate, expand, and differentiate your cells with cytokines, chemokines, and growth factors from STEMCELL Technologies. These reagents are validated to ensure reproducibility across a variety of applications for immunology and stem cell research. Explore more at www.stemcell.com/cytokines.
For the second part of the show, for our interview, I am very happy, and Jason is too, to be welcoming Dr. Adrian Erlebacher. He is professor at the Department of Laboratory Medicine at the University of California in San Francisco. And I'm very excited because during the AAI conference in May, I actually was in one of his talks about reproductive immunology, and I thought it was so interesting. So I'm hoping we're going to talk about some of his the story that he discussed in this presentation, and some other work he's doing in his lab. Dr. Erlebacher, thank you so much for joining us to the Immunology Podcast.
No, it's a pleasure to be here.
Thank you for being here. All right, Brenda, go first, since you saw a talk and are already
yes,
ready to go.
Well, in the meeting in May, so I attended that talk, as I said, and it was very interesting because you were talking, Adrian, you were talking about the trophoblasts and glycans that regulating fetal maternal tolerance in pregnancy, and I thought it was very interesting. I think in general, maternal tolerance, how is it that fetuses survive, and this kind of tug of war between the fetus wanting to survive, the mother's body rejecting it, and how do you find that middle ground? I think it's in general super interesting. So I was thinking that maybe we can start a little bit from the basics. And maybe you can tell our listeners what's the deal? Why is it so, in a way, uh, contradictory pregnancy from an immunological point of view? And what do we understand so far about how this comes to happen?
Of course. Yeah, so the paradox of fetal maternal tolerance is the term that people use, really came about through the work of Sir Peter Medawar, who is the immunologist who won the Nobel Prize for discovering the immunological basis of transplantation rejection and transplant rejection. And he realized early on that, hold on now, pregnancy is this really crazy situation where you have this essentially normally a half related organism within you. And unlike a surgical graft, your body does not reject that. And so he wrote a very famous paper, I think it was in 1953, talking about this paradox. And he laid out three different ideas that he had at the time, all three of which are not really true. We now know, but that has sort of inspired generations of immunologists and the medical students that are in their classes to ask them, why isn't the fetus rejected? And the answer really hasn't been completely solved. I think we've been making some progress in recent years, but it's still a work in progress.
Hypothetical question. So I'm going to go afield a little, but then come right back in. Could you, given what we know today, could you put a kidney in a uterus somehow, and would that protect it from the host immune system? So say your sister's extra kidney or something, right? So same gender, same sex, half genetically identical ish, right? You get where I'm kind of going, like,
Absolutely. No, that's a great question. And you actually brought up the half identical bit, which is an issue that people ask actually a lot. And you don't even have to be half identical for it to work, because women can be surrogate moms, mice can be surrogate moms to embryos that are completely unrelated to them. So it's not the half thing that really matters. But you're absolutely right. So why couldn't you do the experiment? You do transplant. So people did actually back in the 60s and 70s those exact kind of experiments. They didn't know what T cells were at the time, but they asked the same kind of questions that you're asking. So you put a skin graft into a mouse uterus, and can you get it to, an unrelated skin graft, can you get it to survive? It's hard to do those experiments. And my vague memory reading those papers is that it's like you maybe get a day or two, but it's very hard to really interpret them because of the other complexities of the system, including whether the mice actually have to be hormonally pregnant and whether the uterus needs to transform into its specialized state during pregnancy. So it's not the uterus, it has to be probably the pregnant uterus. And it's not the organ. Any old organ probably won't work. Right now we think that it's the placenta that really is where the action is.
Right, because let's think about anatomy a little bit, and how exactly is this contact, right? Because the placenta is genetic material from the fetus, right? And
Yep.
so the trophoblasts, these cells that belong to the placenta, they are in direct contact with the mother's immune system, in a way, with the mother's bloodstream. So, and I know some of your work has been on how do we establish this tolerance, has to do with specific properties of these cells and the way they present themselves to the maternal immune system. So maybe we can start asking, through your work and maybe work of colleagues in your area, what are the mechanisms through which this tolerance is established?
Yeah, that's a great question. And in a way, that the paradox, I mean, you nailed it by talking about the placenta, because in a way, the paradox of why the fetus isn't rejected really is the paradox of why isn't the placenta rejected, because it's the placenta that really forms the interface between the conceptus, which is a term that people use to sort of refer to everything, the fetus and the placenta, and the maternal immune system. And I think what happened in the last 20, 30 years is realizing that there are really two interfaces that we need to be thinking about. One interface is obvious, and that is that the embryo is implanted within what's called the decidua, which is the specialized uterine mucosal tissue that forms during pregnancy. And so that interface is very physical and direct, and the decidua is populated with maternal immune cells. So question number one is, why don't those immune cells interact in a negative way with these trophoblasts that are invading into the decidua and are present right there and living there? And then the second interface is the interface between the maternal blood circulation and trophoblasts. And so here's where the developmental biology of the placenta gets really, really crazy and interesting. And that is, in order to co-opt maternal blood flow so that the placenta can function as its nutrient and gas exchange organ, what happens is that trophoblasts invade into the arterioles of the uterine vasculature. And so essentially reroute blood flow through the rest of the placenta. And so that's an incredibly important process during human development and mouse development. If it's messed up, you have pregnancy failure or diseases like preeclampsia. But what it means is that special kinds of trophoblasts are literally being bathed in maternal blood. And those trophoblasts are releasing material, including antigenic material, into the circulation. And it circulates all over the place and can get, these antigens can be picked up by antigen presenting cells in the spleen, for instance, in a mouse. And so when we think about the problem of fetal maternal tolerance, really we need to be thinking about those two interfaces almost as potentially separate problems. But my suspicion is that there's going to be some sort of grand unifying theme behind it.
So can you take substance from the trophoblast or cell effluent or cultured, media and use that as a general immunosuppressive in, I don't know, an orthotopic graft to pick something that mice don't like and then see them like chill out with it or what have you?
Yeah. I mean, in a way, that is the one of the great goals of the fetal maternal tolerance field. Can we make a transplanted kidney have the same properties of the placenta, either by sort of genetically engineering it or, giving the transplant recipient the magic stuff that's coming from the placenta. So that has not been achieved yet, but that definitely is one of the goals.
So I know this is not particularly your work, but I remember for them same session, some of your colleagues in the field were talking about you could make a bulb C, I think it was bulb C mouse, not reject a black six skin after this bulb C mouse had been pregnant with a black C, a half black six fetus. I think that was a story, something around that you could generate tolerance through pregnancy that then could extend to other parts, other organs.
Yeah, so the postpartum responses of moms to antigens expressed by their conceptors is kind of complicated, actually. There's evidence that you have extended graft acceptance, pardon me, for like skin grafts, but also accelerated rejection for tumors that are strain matched to the father. So it's not. So I don't like to think about it in terms of because I don't think that the evidence really suggests that it's classical tolerance. It's something a little bit more unusual and special, and that's kind of where the edge is right now. So you were asking about like what are the mechanisms of fetal maternal tolerance, and I can go through sort of a short list of various ideas, but the idea is really to focus on or to make a distinction between what's going on systemically with this antigen that's being shed into the circulation versus stuff that's happening locally. So the antigens being shed into the circulation, the sort of the big theories right now are regulatory T cells, and then our more recent work that the antigens are glycosylated and imbued with these sort of specialized immunosuppressive glycans. And then on the uterus side, there's this idea that the uterine stromal cells are immunosuppressive and causing local immune cells from doing their normal thing. So that's sort of like the broad view.
Can you talk a little bit more about the glycosylation?
Yeah, sure. So this was work that was done by an ex-postdoc in my lab, Gabby Rizzuto, who has her own lab at Memorial Sloan Kettering now, if you want to work on this problem in New York City. And so we were kind of stuck in the lab of trying to understand why was that CD4 T cells seem to have a different again, non-immunogenic response, but different than that of CD8 T cells in a system where we knew that the cells were seeing this antigen that was being shed from the conceptus. So we have this experimental model where we express a transmembrane form of, our favorite model antigen, chicken egg ovalbumin or OVA, and it gets released into the circulation, and we can track CD4 and CD8 T cell responses. And the CD4 cells, they see the antigen, they start proliferating, but you can't activate them. Even when you sort of try to juice up the mouse and convince her, with adjuvants, and convince her that this antigen that she's seeing is sort of a dangerous antigen, sort of a pathogen-associated antigen. So, we give the mice adjuvants in mid-gestation, trying to sort of trick her that the placenta is this dangerous organ, and the cells still don't get activated. And so that basic observation led to discovery that the antigen is really has some very specialized properties that acted in cis. In other words, that they were attached, they were physically associated with the antigen itself, right? So, and that came about when she was studying her responses. Gabby was studying the responses to B cells. And so what she found is that the B cells turned out to be the cells that are presenting the antigen to the CD4 T cells. And then in turn, the B cells that are antigen-specific, OVA-specific B cells themselves don't get activated. And so trying to understand why that happened led her to realize that the antigen actually has this N-glycan attached to it that's immunosuppressive. And it's immunosuppressive most likely because it acts through these inhibitory receptors, these glycan inhibitory receptors on B cells that are called Siglecs, in particular CD22 and possibly Siglec-G. So the idea in general is that the trophoblast, and we can see it with this one specific model antigen, gets decorated with this immunosuppressive, highly sialylated glycan that causes essentially any B cell that picks it up and wants to present it to a CD4 T cell to be suppressed at the same time. So it can't do that. And so the B cells get suppressed, and because the B cells are doing the presentation, the CD4 T cells are suppressed.
I thought that was such a cool story. And it's so direct, and I think that I was very impressed by the story, and it's very interesting to see how this telerogenic responses, the cells still proliferate, but there's this separation between like proliferation and the effector molecule production, and it's so cool that this is mediated by these fetal cells directly through this. They're kind of tricking the immune system into developing the cells, the activated cells in this way. um, and I know that you have other work in your lab. So this is the story I had heard, and I'm really happy that you get to share it with your listeners. But I know that I just want to make sure that we also ask a little bit about other stuff in your lab, because that one the other things I'm not so familiar with. And I also thought it was very intriguing, because there's other parts of pregnancy that are in a way mediated or influenced by immune cells that I wasn't really aware of. So some things like, that if I understand correctly, the initiation of labor, or the kind of the end of pregnancy, has also some implications for immune cells. So maybe would you like to just give us a little bit of a flavor of what other unexpected parts of pregnancy are influenced by the immune cells of the mother or the fetus, or the combination?
Yeah, absolutely. That's a great question. So so this relates actually to the maternal fetal interface itself. So the question that you're asking in a way is, to what extent does the immunology of pregnancy influence pregnancy outcomes like preterm birth or pregnancy failure? But conversely, to what extent does the specialized environment of the pregnant uterus control immune cell behavior? And I think that there's this two-way dialogue that's going on, that's happening, that scientists are only now beginning to really start teasing apart, and starting to think about how like developmental problems could lead to immunological problems, which could then feed back onto pregnancy outcomes in sort of a complicated back and forth. And so, you alluded to preterm labor onset, and the clinical problem of preterm labor, and that A lot of scientists in the field are really thinking about how inflammation and immune cells locally within the uterus contribute to labor onset. One of the major reasons for people even thinking about that is that a major cause for preterm labor in humans, and I should emphasize, preterm labor is this huge clinical problem. It's, 10% of all pregnancies have experience preterm birth and preterm labor. There's really, there's essentially no treatment for it, something that it can extend pregnancy for a few days or so, then that's about it. And and so it's long been appreciated that one cause of preterm labor is infection of what are called the fetal membranes, which are one of the sacs that are around the conceptus. And because of that inflammation, immune cells get activated and produce pro-inflammatory cytokines. And for reasons that we don't really understand or cannot completely understand, that leads to that trigger, that magical trigger that happens at the end of gestation, where what was previously a quiescent, smooth muscle tissue suddenly transforms into something that's highly contractile. And so trying to understand how the immune system contributes to that is not only important because of this problem of preterm labor, maybe you could ameliorate that trigger, but also the hope has been that by understanding how the immune system is feeding into this contraction pathway, we'll gain insight into what's naturally inducing this contraction pathway, potentially in the absence of inflammation. With the idea that there's sort of like a common downstream pathway. And the reason people are interested in that is, this aside from immune contributions to labor onset, really nobody knows what is the ultimate triggers that transform the uterus into this contractile organ, and also like what times out, is there some sort of timer there that says, okay, 9 months, time to go, time's up. So that's one big area where sort of people are really thinking about how immune cell behavior at the maternal fetal interface can contribute to pregnancy outcomes. Then the other way that people have been thinking about it, and including ourselves, is early on in pregnancy. So I alluded to this problem of how do you attenuate immune cell behavior at the maternal fetal interface so that you don't reject the fetus, sort of that other interface that we were alluding to. But so how does that happen is one big question. We think it has to do with the stromal cells, the specialized properties of the stromal cells that they generate in early pregnancy. But also it raises questions like, well, how do you have infectious disease control at the maternal fetal interface? Because it's almost, you don't want to just sort of say nothing can happen here, or else you're just sort of a setup for infection and congenital infection. But in fact, you kind of are a setup. There are a lot of infections that can sort of, set up shop, like listeria is a great example, can set up shop at the maternal fetal interface, even though in the pregnant uterus, even though you can fight listeria just fine elsewhere in the body. So that's the other sort of major area I think that the field has been focusing on, is like, what are the specialized behaviors of immune cells at the maternal fetal interface and the crosstalk between those cells and the stromal cells, which sort of seem to be the orchestrators.
Do you think this is why like you mentioned listeria, but also why some of these viruses have such profound effects in fetuses as opposed to adults, where you'll just get a little bit sick and walk away, but in the fetus, like, it rages and does something very different?
Yeah, absolutely. So the idea has been that that the decidua, and that specialized endometrial tissue, tends to resist being colonized by a virus or a bacteria. And similarly, the syncytial trophoblasts of the placenta resist being infected. But once you get through that initial wall, then it's because of the specialized or suppressed immune properties of these tissues, all hell breaks loose and some really bad things can happen. So it's like evolutionarily, there's been these like sort of the idea right now is that there are these innate mechanisms that have been established that essentially compensate for the fact that standard immunology isn't quite working the way it normally would.
So you have a really big wall, but no guards patrolling past it, so to speak.
Yeah, that's a great analogy. Yeah.
I guess that maybe many of, especially if you're thinking about viruses, all the interferon responses that you see normally, they might be a bad idea in a developing fetus, and you don't want that. And so you kind of
Well, that's even another layer on it. So that's something that we are recently very interested in because you would expect, forget about fetal maternal tolerance just for a second, where you have like T cells that are specific for the conceptus or for placental antigens, and they call them causing problems. What happens if you just have a tissue resident memory T cell that was established in the endometrium as a result of a prior COVID infection or influenza infection? And the T cells set up shop there. And then a woman becomes pregnant, and then she gets the flu again, and some antigens, you somehow reach the uterus, and those cells get activated again. Exactly what you're saying, that, just pro-inflammatory cytokines, that could be bad. And so that's another, I think, a big, related question. And that is, to what extent the early pregnancy failure or pregnancy problems, like preeclampsia or intrauterine growth restriction, to what extent is that due to non-antigen, well, antigen driven, but the specificity of the T cells that are responding are not to the conceptus, but to something else, and essentially a bystander damage. This is, pregnancy is a situation where everything's got to work right, and even
Yeah.
just low level damage in a short period of time can have profound effects.
On the one hand, you've just described a whole bunch of what we didn't know in the 60s that we do know now about this, and how, just the initial misconceptions being out there. Where is the big barrier still, you think, at our understanding? It sounds like, hey, we're modifying antigen. You have some really innate resistance plus a general likely Treg mediated suppressive environment on the inside. So, walls and no soldiers. But where's the big mysteries on this?
Yeah. Well, our glycan work was only came out 2 years ago. So, as all good scientists, we would love to see this validated in other different ways. One big question is to what extent this is applied to other antigens expressed by the placenta. To what extent could such sugar coating affect immune cell behaviors locally within the uterus? So this is sort of getting to the sort of the grand theory. Maybe it all has to do with the glycans or a large part of it, but we don't know that. And then I think that one of the really big questions how do we translate what we're starting to understand more in mice to humans? That's a really, really big difficulty, because obviously we can't do invasive experiments in humans. We can get blood, and we can get, the products of conception, we can get fetal membranes when the placenta is delivered. But it's very, very difficult to study this in humans. And so, my hope has been that we discover something in mice, and that sort of allows us to laser focus onto some very specific thing to test out in humans and see whether we can see it there as well. Especially so what you're also raising is especially the case when we think about studying causes of labor induction, parturition, because mice and humans go into labor hormonally in a very different
Mm.
place, and they're very different. And so it's been very difficult to translate what's going on in mice to humans. For instance, you knock out all these genes that knock out big parts of the immune system in mice, and there's no parturition, no labor onset problems at all. And so the question is that maybe the immune system doesn't have anything to do with human parturition, or it's because mice go into labor because they stop making progesterone, which is a really key pregnancy hormone. And instead, humans go into labor still with high serum progesterone levels. And so that basic sort of reproductive endocrinology is so different. And that's created a big, big problem in the field, because because we really want to understand what's putting women into labor. But how do you do that if you can't study it in mice?
Yeah, that's very tricky. So, for example, when it comes to the glycans and the differences that you see on trophoblasts in mice, have we been able to look into glycans on human placenta?
Oh, we're doing those experiments right now. Yeah, exactly. Can we find evidence that they're decorated in the same way?
Okay, so you're
And
looking into that.
yeah.
Because I just want to say, not to go too deep into that, but one thing that I didn't know before attending this talks on the AI is that there's so many variations in the design of kind of the placenta and the uterus between different species of mammals, and that we have sometimes the placenta is not in contact with the mother's blood in other kinds of animals. I think was like, I had my notes, there was cows and dogs. They're completely different in the way they interact with the maternal bloodstream and immune system. So it must be also very interesting to think of the implications of how different mammals have, found ways to respond to this paradox of the fetal maternal interface.
Yeah, no, it's a really interesting question, on the one hand, it makes it very interesting with the divergence in reproductive strategies and the rapid evolution of these different forms of placentation have have caused a big problem. So ironically, given how this difference in the endocrinology that I alluded to before between mice and humans, one thing that is in common is what's called the hemochorial structure of the human mouse placenta. So you're right. So in other species, there's not this cooptation of maternal blood through the core of the placenta that happens both in mice and in humans. So, antigens are not released into the circulation as far as, like in a pig. So that's been a lucky break for us. There's one thing that is in common between mice and humans.
Yeah. What are the odds that you can actually
No,
use
no,
it?
seriously, they're very, very different. Yeah.
Interesting. All right, so we're going to have to wrap up here, but I had one career question before we switch to some fun questions, which is you're in the Department of Laboratory Medicine, Pathology Department. Everyone has a different name, slightly different name for that thing. But you study OB work essentially, and you're an MD PhD. So did you end up doing like a residency in pathology and ending up in, figuring out rejection, which led you into, pathology, which leads you into this? Or did you end up in OB, which led you into not rejection and leading you into this, or option C, which is not one of those? I'm just very curious as a physician scientist.
Yeah, none of the above. So I didn't do a residency actually. So I don't see patients. And I got interested in fetal maternal tolerance when I was coming out of my MD PhD training and deciding not to do any clinical work. So what department does somebody like me wind up in? And when I started my lab at NYU, I was actually in the pathology department, at UCSF, pathology and laboratory medicine are separated departments in many
Ah,
ways. They're combined. So sort of along the medicine, the pathology lab medicine track, but I wouldn't have to be in that department. And the reason I am is because my chair, Cliff Lowell, recruited me to UCSF, and he is in laboratory medicine. So this in a way is one of the really great things about UCSF. Departments in terms of your intellectual community don't really matter so much. So I'm in laboratory medicine, but my sort of intellectual home is the immunology program, the ImmunoX program at UCSF, as well as the UCSF Center for Reproductive Sciences. And so those are some of them are in lab medicine, many of them are not, but in a way, it doesn't really matter.
yeah. I was just curious, assuming the clinical part in there, but yeah, you chose wisely in some way.
You make your own path. But that's so well. I think that as we are wrapping up the conversation, we're gonna ask you a little question. To get to know you a little bit better, for our listeners to get to know another part of your profile. So a question that we ask very often is, if you were not a scientist, what do you think you would end up doing? And we talked about it a little bit before starting the recording. So why don't you share this with our
Yeah.
listeners?
So when I was younger, I always was doing science, but I wanted to be a composer at one point. And when I was in college, I was writing a lot of music, and I was heavy into the classical music scene. And I was very lucky. I was able to take a year off after college and study music full time, and that was great because it was a fantastic year. But it was also very good because I realized I didn't have the talent to become a composer. And it was just a really, really good thing to realize early on. And so luckily I had science still was percolating along, and I was already going to be going to medical school and MD PhD school. And so I continued along the parallel track and wound up where I am now.
So how does it work? Because you were already accepted to med school and you decided to take a year for composing music. How does this work from a person that has never been in that system? How is that
Oh,
possible?
yeah, I was very lucky. So I actually did my MSTP training, medical sciences training program at UCSF. So I'm back in my home after, going back to the East Coast and doing postdoc there, et cetera. And I received this fellowship after I was accepted, and you know, um, the director of the program at the time was very gracious and said you could defer for a year. So I'm very grateful.
That's very nice. That reminds me of in my undergrad, we had one of my good friends. He I studied biotechnology from my undergrad, and one of my good friends there, he joined us in the first year after having been in the conservatory for a whole year studying classical guitar, and then he decided that he could not see himself doing this for the rest of his life. And it's funny. He's like, "Well, I'm not doing classic guitar. Let's just do hard science." And he was one of the best in our class. Definitely, he was like just sweeping the floor with all of us. And he's like, "Yeah, I just figured I didn't want to play the guitar for the rest of my life, so I wanted to do something a little bit more meaningful." And so, it's so cool to see people just, you can be fluid. You can, to get a little bit of both worlds. I was very impressed by my friend.
One of my fellow med students was on Survivor.
That's also very
Wow.
impressive. Sorry, and did he survive?
He didn't make it to the end. He made it decently far and then didn't. But yeah. Yeah, no. Do you compose at all now, or just write a little bit on the side, or dabble with instruments, or?
Yeah, well, I watch my kids become musicians, which is really
Oh, there you
just
go.
so.
All right. Well, on that high note, pun intended. I think we want to really thank you, Adrian Erlenbacher from UCSF, for joining us today at the Immunology Podcast. It was very interesting. I was really happy to talk to you after being very much impressed by your talk at
My pleasure.
AAI.
Thanks so much for having me.
Thank you. All right. Well, that brings us to the end of our show, folks. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on X at @immunopodcast or via email info@immunologypodcast.com with feedback or to suggest guests. See you next time.